post. I'll come off with that. You're the only person there on Vision.
There's a few.
Hi, good to see you.
David.
Hi. Hello, David.
Looking very smart, Ahmed.
Yes,
Good to see
thank you.
Yeah.
Thank you. Thank you. I am really very honored and glad to be among you today to start the fourth seminar webinars of ASCO International Webinars. As we agreed together before that these webinars are recorded to be part of a complete series of 5 webinars addressing how ways to game changing in global oncology. So it is not the number, the classical part of the classical way to have some webinars of some audience, leave audience or something, and goodbye after. But it's something continue. This is the value of this webinar is to it's record, it's material to be part of a movement. Also, it is a part of a certificate of game changer in global oncology for our mid-career and younger colleagues. We send for them all material, all the past recorded webinars, and these webinars and the upcoming webinars also. And we send you examples of evaluation form and their comments, because it is very important that this message be led and be continue by generations and generations. You are the leaders. You are who raise the flag in front of all, but they should be adopted and be part of practice and research and a big field of research. We had all of us had enough of slogans and about publication, hundreds of publication about disparities and about equality and about donations, about charity and about global fund, but the outcome, as you know, after all these decades of organizations and everything and all our efforts, we are partner in this. We are included in all this. That the gap between, as you know, all better than me, that the gap between what is required and what is available is widened, and it will increase, and it will be with skyrocketing. The gap is increased despite of all sessions about disparities, equality, and about how to think wisely and how to think of the only way that we have as professional is your wide experience, this treasure of experience in how to use science in research in getting better value cancer care, as always Professor David Kerr said, and we learned a lot from publications and work of many. Like we have an honor today to have Professor Ian Tannock. Personally, I learned a lot from him. If you revise our last book together, you will find many references coming from Professor Tannock and Professor Rattan and Lister and surely Professor David Kerr, Professor and Sir Moore Gray about triple value. So all this should be translated and gathered together into a movement, a win-win movement. All would win. If we continue as we are with talks, seminars, and all these things, I think that the system will collapse. We should scientists should do their roles in giving how to how to reduce enormously the total cost of treatment without compromising the outcome. It is not like, as some of us say, do with what we have, or or some essential drugs, and this is the solutions. This is non-ethical even. Okay, to continue like this, but we can give. It is a big challenge. It is difficult. Yes, some say this is dreams. This is reality, not reality. No, it is reality. It is by science, by you. I am just. I learn from all of you. By the way. Today we'll start our fourth and fifth. This is continuous. The fifth. Today is the fourth webinar. In 18 June, this is the fifth webinar. It is continuation of the webinar of today, but we'll give more ample time for discussion and for conclusion, or for having reports and something for publication led by you, by these distinguished leaders and faculty, to own the title of this first and fifth webinar is examples of how to decrease the total cost of systemic cancer care without compromising the outcome and to get better value. I give you just a kind reminder that the next series, it starts in September, will be 5 webinars about radiotherapy, radiation oncology, radiomolecular radiotherapy with immunotherapy and nanotechnology. 5 webinars from September 25 to 17 June 2026. There are many brainstorming things. It's not just repeat the same thing in about radiotherapy and about many things. It's a system will collapse, ladies and gentlemen. So this is your big role, and big. We have the honor to present you today, because this is your big role to give scientific way and to push scientific simulation for hundreds and thousands of valid research now in the future. Now we start with just introductory speech, short introductory speech of the president of ASCO, or this president of, this is introduction of the whole series.
Greetings from Niles, Michigan. I'm Robin Zahn, President of the American Society of Clinical Oncology. Today, I am pleased to welcome you to the second installment of the ASCO GOIS webinar series. As an international organization, ASCO is proud to have over 50,000 members in more than 170 countries. Given our own commitment to global impact, it is only natural that ASCO would find such an ideal partner in SEMCO. Since its founding in 2002, the South and Eastern Mediterranean College of Oncology, or SEMCO, has been an impressive champion of professional development and education for both oncology professionals and the public across not only the Mediterranean, but the world. SEMCO has served the global oncology community as a facilitator of collaboration and cooperation between regional and international organizations and societies, including ASCO. This year, it is our great pleasure to partner with SEMCO on the ASCO Global Oncology Implementing Science, or GOIS, webinar series, a complement of virtual education sessions designed to inform cancer care professionals of hot topics in global oncology and explore the scientific and patient-centered approaches to finding and validating accessible and affordable therapies. The goals of this series will be met through a variety of short presentations and panel discussions with international experts during each session. The first webinar in the series, Improving Access to Global Oncology Care Through Repurposing, was a great success, prompting new and interesting discussions on the idea of repurposing oncology medications. Today's webinar will address the myriad issues surrounding implementation of the science of global oncology. We hope that you will find the presentations informative and the panel discussions engaging. Finally, I would like to take this opportunity to thank Dr. Ahmad El-Zawawi for his hard work and dedication to this webinar series. Dr. El-Zawawi is, among many things, the co-founder and co-president of SEMCO, chair of the Win-Win Initiative, and president and founder of iStock. I and all of us at ASCO thank all those involved in the creation and implementation of this wonderful and very important series. We look forward to future sessions and the discussions as they advance. Thank you and enjoy this webinar.
Sorry, Ahmed, I don't think we can hear you. I don't know if there's maybe a sound problem at your end. Ian, I assume that you can't hear Ahmed either. It's not just me, is it?
No, I
You.
cannot. He
I've
was
gone
he was
deaf in
fading
old
in
age.
and out. Yeah.
Ahmed, you're on mute.
It's off mute now.
Hello, I'm David Kerr, Professor of Cancer Medicine from University of Oxford. So let me bring you warm words of welcome from our dreaming spires. You can see the rather beautiful city of Oxford and its ancient university laid out behind me. We're over 900 years old, so we're one of the oldest universities in the world. But that doesn't stop us thinking about how we can do better with the drugs, the treatment that we already have. Oxford is one of the best medical schools in the world, of course. We have some fantastic scientists, Nobel laureates, people pushing at the very cutting edge of science, looking for new treatments, trying to better understand the biology of cancer. Of course we have. But within that, we still consider the win-win promise, the idea to seek better value cancer care that my dear friend, Professor Ahmed, has discussed and put forward during this course is terribly important. Can we do better with the treatments that we have available? For me as a medical oncologist, can we teach old drugs new tricks? Can we squeeze every last bit of value out of the drugs that we've got? And I've got to say that Ahmed has been one of the world's great proponents as to how we might be able to do that in his Win-Win Initiative. And this is based on science. This is based on understanding pharmacology, understanding drug delivery, and understanding, above all, dosing. If you think about how we treat cancer patients, although we've made great advances in terms of understanding the biology of cancer, the genetics of it, we cannot control the somatic, tumoral, mutational landscape. It is what it is. What we can control, though, is how we deliver the drugs, the treatments, the radiotherapy, even the surgery that's available to us to make it as effective, but also as cost-effective as possible. And therefore, by understanding better relating drug concentrations to effect, by revisiting phase 1 trials, for trying to understand what the dose-response relationship is. I think that we could do much, much better. Our dear friend Ian Tannock from Canada has done some great work in this field recently. He published some work looking at the dose-response curves for immune checkpoint inhibitors and showed, I think convincingly, published it, that we can probably, for the doses that we're giving just now, we could probably treat 8 patients instead of 1 without losing any form of efficacy. So that sort of sense that we've got to treat at the maximum tolerated dose, we've got to give the maximum dose, often isn't the case. And by stepping back, reviewing carefully the phase 1 data, looking at what dose causes the best effects, we can get more out of the drugs that we use. So plausible, scientific, evidence-based. I'm still interested in the concept of metronomic chemotherapy, low-dose oral chemotherapy, that again is plausible. For those of us who were brought up understanding the cell cycle, the various phases of DNA synthesis, we know often that prolonged exposure to low concentrations is more effective than a short, sharp, high-dose bolus. And the chemotherapy is much better tolerated when given in that way. Ahmed will give you many other examples as to how, as a community, we can do better with the drugs that we've got. Now, of course, I encourage you to take part in the sort of research that we're involved in Oxford, moving the science forward. But at the same time, I think that we should be doing the best we can and to explore in a systematic way how we can get better value out of the cancer treatments that we have available. Thanks for listening, as always. It's great to be a part of this course in a very small way. And again, 1,000 congratulations to my friend Ahmed for his tireless work, his extraordinary energy, and the fact that he flies the flag for his win-win initiative better than anyone I know. I hope you enjoy this important congress, this important series of seminars. Thank you very much.
Thank you, David. Thank you very much. We have the honor really to that with your presentation, and you will have another one. This is 60 minutes, another one with more details. And now I have the honor to present Professor Ian Tannock. Please introduce a few words in the beginning, and we have the honor of this, and then you will show your very valuable video. Professor Ian, do you hear me? Professor Ian Tannock, do you hear me?
I can't hear you. You're breaking in and out. I cannot hear you. You are saying something.
You hear me now?
Sound. I think it's on your end, not mine. I don't know. I can hear you now, but I don't know.
So we
Thanks, Ahmed.
you hear me, David Kerr?
We can, Ahmed. I think, Ian, I think it might be you that's breaking up this time, bizarrely. You were very, very clear earlier on. I think you've unfrozen. I think you're back in the land of the e-living, as it were. So I think we're all connected up now, hopefully.
Yeah, hopefully. I'm in my apartment in a study, and today is when we have our cleaning lady come, so I can't really go out and sit right above the server. Can you hear me now?
We can.
Yes, we hear you. Yes, we hear you.
I'm not sure what you want me to say. I have been involved with a number of organizations, including the Optimal Cancer Care Alliance and Common Sense Oncology, and a couple of the
We have signals now that
trials
yes.
that give us free of bias and to strongly criticize. Can you hear me?
Yes.
Yep.
Could we show your video? Maybe it is better because it is.
Yeah, I think you should show my video.
Oh, okay.
Good day. My name is Ian Tannock, and I'm a retired medical oncologist from Princess Margaret Cancer Centre and the University of Toronto. And I'm going to talk to you today about using either lower doses or shorter courses of anti-cancer drugs to try to improve their access, particularly in low and middle income countries, and ask what we need to do to change practice to make that happen. I don't accept payments from pharmaceutical company, and I try to preserve my independence. Now, a major problem is that most phase 1 studies of targeted agents really haven't changed since the days of chemotherapy. For chemotherapy, it was probably appropriate to assume that the higher the dose, the greater the likelihood of effectiveness, and that dose-related acute toxicity was a surrogate for tolerance. So the assumption that the maximum tolerated dose is the optimal dose may have applied to chemotherapy, but it really doesn't apply to many biological agents. What we should be doing is for looking at the optimal biological effect. If you exceed the dose of a targeted agent that maximally inhibits its target, all you'll be doing is adding toxicity, off-label toxicity. Now, reducing registered doses and schedules has traditionally required proof of identical outcomes in very large non-inferiority trials, and certainly the pharmaceutical industry, has certainly encouraged that. Now, we've challenged that concept in a couple of papers. One where we've talked about near equivalence, and another where we've tried to attack the tyranny of non-inferiority trials. And I'll try to show you what we mean by that. So the problems of non-inferiority trials are, I think, multiple. First of all, they demand tight statistical boundaries for a single positive outcome. But we're not really interested in a single positive outcome. Sure, we want to make sure that lower doses or reduced schedules of a drug give at least approximately similar outcomes, positive outcomes, such as overall survival or delay to progression. But we're also interested in toxicity, and particularly in low and middle income countries where these agents are often not available because of cost, we're interested in access. So the non-inferiority trials relegates these other outcomes, and yet they're equally important. It often ignores supportive pharmacokinetic and pharmacodynamic data. So if you've already shown that a targeted agent maximally inhibits its target at a lower dose, you should use that information as well as the information you
have to come here because our internet is not working.
And comparison of outcomes within a non-inferiority trial doesn't actually allow that you'll get a variable result. It's almost you do one superiority trial and then you regard the result of that as absolute, and a hypothetical trial might not even show standard treatment to be non-inferior to itself, which of course is nonsense. We think that differences between superiority and non-inferiority trials is artificial, and it also depends on when a trial is performed. You do an initial trial to show a new treatment is better than a standard of care, and that requires maybe a few hundred patients, but then you need a much larger trial to show non-inferiority of reduced dose, and that makes no sense. So what we propose is to abandon the term non-inferiority trial. We think all randomized trials are simply comparative. The preferred arm should be selected on the basis of survival outcomes, toxicity, tolerance, and cost. And comparison of registered and reduced doses and schedules should take into account all available information. Smaller randomized trials, but also pharmacokinetic and pharmacokinetic data should be taken into account. And interestingly, that is done when we register biosimilars. So many randomized trials have actually shown similar or identical survival for lower doses or lesser schedules, but those regimens have been rejected despite reduced toxicity. And here's a few examples. So here's duration of adjuvant trastuzumab, 1 year versus 6 months, or 3 trials of that. 1 year versus 9 weeks, 2 trials of that. Look at those survival curves, they're superimposable, and these were large trials. Surgery for colon cancer. Is laparoscopic surgery better than open surgery? Well, they're the same. Supportive care in non-small cell lung cancer versus radiation for brain metastases. Or continuous or intermittent androgen deprivation therapy for prostate cancer, or duration of adjuvant FOLFOX or CAPOX for colon cancer, all of them giving survival, or in the case of the colon cancer, relapsed disease-free survival that are essentially identical. So here's looking at 2 of those very large trials of adjuvant trastuzumab, one with 6 months, one with 9 weeks. Both of them concluded that non-inferiority of the shorter schedule had not been established. But clearly they give the same outcomes, and the advantage of lesser treatment is clearly obvious. It's much cheaper, therefore affordable, more affordable in low and middle income countries. And although trastuzumab is not a very toxic drug, it's less toxic. And in fact, if that short hair trial of Conti et al. Had been done before the HERA trial, which looked at 1 and 2 years, 9 weeks of trastuzumab would currently be our standard, except not a year. And yet everybody, or almost everybody in the Western world, still gives a year of treatment. And then why do few patients in low and middle income countries actually receive immunotherapy? Now, the 2018 Nobel Prize in Physiology and Medicine was given to Jim Allison and Tasuku Honjo for their discoveries in cancer immunotherapy, highly appropriate. And these drugs were developed in part with research funded by public funds. But except for treatment of melanoma, these drugs are not on the WHO essential drugs list because of cost, and they're not available to 80% of the world's population. A simple question, and I think you can all answer that question. Surely this is not an ethical state of affairs. And there's plenty of evidence that we can reduce the dose or schedule of almost any of these agents. Here's some data from nivolumab. This is the PD-1 receptor occupancy, so that's the target, on T cells at 8 weeks after various doses. And you can see at even the lowest dose, which is far less than that registered, there's essentially the same binding. And then this is a large phase 2 trial of 168 patients for renal cell cancer, looking at 3 different doses of nivolumab, 0.3, 2, and 10 milligrams per kilograms every 3 weeks. You can see overall survival curves are not different, nor is response rate or progression-free survival. And yet what happened? The registered dose of nivolumab is 15 times the lowest effective dose in this trial. And if you actually look at the data from pharmacodynamics and pharmacokinetics, and there's new data coming out, you could probably reduce these doses and hence the cost up to somewhere between 85 and 95%. And that would enable you to treat 7 to 21 times as many patients as we do with a single dose. Of course, that's fought tooth and nail by the companies that market these doses, but it is beginning to happen in India, in other low and middle income countries, thereby enabling patients to have access to what are still effective doses of these drugs. A practical trial that might convince governments, oncologists, payers, patients to use low-dose immunotherapy might be a simple trial of, say, patients with lung cancer, a common disease with a high PD-L1 score, and randomize between the registered dose, in this case pembrolizumab 400 milligrams every 6 weeks, that's equivalent to 200 milligrams every 3 weeks, and 100 milligrams every 6 weeks. Most of the trials are looking at different schedules, which is harder, but in this case you could actually do it double blind, and that would, I think, be a very good way of doing this type of trial. And the major challenge, of course, is how to pay for the pembrolizumab, especially the full dose, because the company isn't certainly going to pay for it. And what about other drugs? Abiraterone is a very effective drug that's improved survival for many stages of men with prostate cancer. And this was a near equivalence trial, only 72 men randomized to receive full dose, registered dose abiraterone on an empty stomach versus a quarter dose after food. And there was plenty of data showing that food increases the bioavailability by a factor of about 5 to 7. And not surprisingly, if you look, admittedly at an early endpoint, at change in PSA at 12 weeks, there's no significant difference. There's even a greater trend for a greater fall in PSA in the low-dose arm, and the time to PSA progression is the same in both arms. The blood sees the dose, the tumor sees the dose in the blood, not what you give to somebody in their mouth. And the similar PK, the similar pharmacodynamic effects, and we did actually apply successfully, Len Saltz and I, to NCCN to include low-dose abiraterone with food in their guidelines. In another study with a colleague, Amol Patel, we've also shown that adoption of the lower dose abiraterone in India can lead to a saving of about $180 million a year for that low dose alone. And that is being done now in many lower middle income countries. So there are absolutely many drugs that are given at much higher doses than are needed. And these are a list of some of the drugs where there is strong evidence that you can use lower doses and thereby make them much more affordable and much more available, particularly in low and middle income countries. We do know, of course, that the US FDA has introduced Project Optimus. This is a good thing. The FDA now requires dose optimization studies for new drugs, but it doesn't apply to drugs that are already approved. And one example, sotorasib, which is a KRAS inhibitor for KRAS mutated lung cancer, non-small cell lung cancer, it suggests that dose optimization is not being applied critically. The FDA required Amgen to conduct a dose-finding study of 960 versus a quarter of that dose, 240 milligrams. There are the results. There is no difference in either progression-free survival or overall survival, but a substantial difference in toxicity, not surprisingly, that 240 milligrams is less toxic. But despite this lack of significant difference in outcome, and I think we can see the outcomes are extremely similar, but greater toxicity, the FDA has still gone ahead and allowed Amgen to endorse, or has endorsed the use of the higher 960 milligram dose. Our system is broken. That should not happen. So we need trials, pragmatic trials, evaluating less intensive therapy. There is a false requirement for them to be large non-inferiority trials, and I've already addressed that. There's difficulty of funding them. It's not a sexy thing. People like to give their money to new treatments. It's, I think, an inappropriate focus of funders, journal editors, and reviewers on one outcome measure, progression-free or overall survival, with neglect of benefit due to reduced toxicity and cost, and hence greater uptake. And there's a great difficulty in stimulating uptake of lesser treatments, even with this strong evidence to support it, as I've already shown in an earlier slide. Pairwise comparison. This is an interesting statistical method of comparing lesser treatments with the current standard of care. Led by Mark Boyce and Michael de Backer in a couple of papers here. How is it done? Well, in the pairwise comparison method, each patient in the experimental group is paired with each patient in the control group. A relative score is assigned based on relevant outcomes. So you can compare every patient in one group with every patient in the other by computer methods. The score is +1 if the experimental group is better, -1 if the control group is better, and 0 if the outcomes are judged similar. And then you simply add up the total score, summing over all the pairs. The method requires individual patient information, so it's very good moving forward for future trials evaluating reduced dosage. So, in conclusion, many anti-cancer drugs are used at higher doses and more frequent schedules than are needed to fully inhibit their targets. Pragmatic trials to evaluate dose optimization are important. Near equivalence of positive outcomes, such as overall survival, with reduced toxicity and cost, should be used to decide the optimal dose and schedule. And any questions you'd like to send me, please send me an email to my email address given there.
So I've moved now to be near my server because I think the internet in the other room was breaking up. So if people have any questions, I'm very happy to try to address them.
Allow me, please, to say something. This is your lecture is like a pearl. All your works, I learned from it, from them, many. But the question is, and I would like to ask Catherine later on, when after David Kerr's speech and her speech, why this very needed work and scientific work are not sexy, as you said, or let us say, not attractive. While if you have these webinars in pharmaceutical company in my country or other countries with pharmaceutical company, you will find full theater, full and a good dinner after and accommodation in hotels and all these things. And what is the message there is to use expensive drugs without giving scientific evidence. But we are patient and we are with you and behind you, all leaders, to attract more and more people. Don't be look for the number of online today. It is 22, but the registered in GOIS till now is 400 till yesterday evening, 420 colleagues from all over the world. All of them, we are very patient. We send for them records. We give them evaluation. We ask them to lead. We ask them to publish. And we encourage e-cancer and global oncology and all of you to encourage them to publish something, things about, driven from your pearl or your this is really pearl lecture. So we are online all the time. It is not just the webinar now. I am very grateful to Professor Ian Tannock, and thank you for giving us your email, and we'll be in contact, and your leadership for this movement in the world with some patients, I think that can change the game in oncology. Thank you. Thank you. Now, I'll give the talk to Professor David Kerr, and we'll have also another pearl lecture from him for 60 minutes. And we have Catherine, very important speech from Catherine Van Loon, the editor-in-chief of Global Oncology Journal, GCO, and it is very important speech, really. Now, David Kerr, introduce a few words, and then we'll run your video.
Oh, great. No, just to reflect on what Ian was saying, it was a fantastic talk, it really was. It made me smile. In our field, colorectal cancer, we did a 12,000 patient non-inferiority trial demonstrating that 3 months adjuvant chemotherapy was non-inferior to 6 months. 12,000 patients later, multi-million dollars spent in terms of trials. There's still a debate about it in terms of subtle variations of chemotherapy in staging. So it's remarkable to me the degree and quality of evidence that we require to supply to get some of our colleagues to change position. It is genuinely remarkable. I don't know, Ian, if you and I, as relative oldsters, you and I have had this conversation before, are too easily convinced. But 12,000 patients, a remarkable international cooperative effort, and still there are question marks about non-inferiority. There's something psychologically very, very interesting about it. But it was a fantastic talk, so thanks for that. No, Ahmed, if I made a video, I don't recall, but if I made a video, fantastic. Push the button and let's roll.
Okay.
Cool.
Okay.
I thought I was going to have to do something live, such as my memory, but
No problem. We organize everything.
hooray! That gets me off the hook. Well done. Hello, I'm David Kerr, Professor of Cancer Medicine from University of Oxford, and what I'd like to discuss today, this excellent series of webinars that my great friend and colleague Ahmed Ozawi has arranged, is to talk about optimizing dose selection for cancer medicine. You can see behind me the Dreaming Spires of Oxford, and I first came to Oxford as Professor of Clinical Pharmacology and Cancer Medicine. And what I hope to be able to do today is to be able to convince you that it's possible to use some simple scientific principles to optimize the dose of drugs that we deliver. Because when you think about it as oncologists, that's what we can control. We cannot control the genotype we deliver cancer medicines. So these are the topics that I'd like to discuss a little in more detail. Classical phase 1 trials to determine the maximum tolerated dose. These are something that I started doing 40 years ago. And basically the essential trial structure hasn't really changed since then. Using a thing called an arithmetical progression called a Fibonacci scale, we treat 3 patients, we double the dose of the drug, we treat 3 patients, we double the dose of the drug until we see side effects happening, and then we increase drug dose by smaller increments until we reach a ceiling, so-called maximum tolerated dose. So the idea is that we continue to dose to the point at which the patients can tolerate the treatment no longer. A very classical design, and one which has been used since the beginning of experimental chemotherapy. It's an important step from the laboratory bench to the patient's bedside, in which we take novel chemicals or gene therapies or vaccines or tyrosine kinase inhibitors, whatever the mechanism of action, they still go through this rather elementary toxicology-driven phase 1 trial design. Problem is that it's statistically very unstable. There are only 3 patients per dose level. And then when we get to maximum tolerated dose, there may be 6 or 7 or 8. So think of that. We may be basing the treatment of thousands, tens of thousands of cancer patients, millions even, and a dose-finding schedule that may be based on a n equals 3. So a degree of statistical uncertainty, to say the very least. Not that that's caused us to change. It may be better to use Bayesian forecasting techniques, and there's some interesting ideas going on there. But the fundamental structure of how we introduce new drugs to the clinic hasn't really changed. people have thought of modifications over the past 40 years or so, and we've been at the forefront of testing some of these different approaches. Pharmacokinetics, so we measure, it's what the body does to the drug. So we measure the drug levels in the bloodstream, and we hope that we can define a therapeutic window. By that I mean there's a lower, a minimal plasma concentration that we want to achieve, because below that the drug would be ineffective. There's a maximum plasma concentration we want to achieve, because above that the drug will be very dangerously toxic. And between these two levels, we have the therapeutic window. So in an ideal setting, we would adjust the dose of the drug to achieve a preset plasma blood concentration level that gives optimal efficacy with tolerable toxicity. Problem with that approach, when we tried to apply it, was that, and because of intra and inter individual variation in pharmacokinetics, how we metabolize the drugs, how we excrete them, the variation was very, very large. So in a phase 1 trial, trying to dose escalate on the basis of plasma concentration proved impossible. Many of us tried, all of us failed. And that idea about defining a therapeutic window for anti-cancer drugs, attractive though it is really difficult. Many of our drugs have got irreversible binding kinetics, and that makes it very difficult to build an appropriate mathematical model. Pharmacodynamic endpoints, that's when we try to titrate the biologically effective dose of the drug. Say we're inhibiting a particular kinase, can we, during the phase 1 trial or phase 2 trials, can we collect blood samples for pharmacokinetics, and can we collect cells that would allow us to see if we're inhibiting the target enzyme, if enzyme it is, to the right level? And we can do this using tumor biopsies. Difficult to do that frequently, but sometimes we use surrogate tissues, skin biopsies, collecting mucosal cells from the lining of the mouth. I remember once as a research fellow plucking eyebrow hairs, and if you do it properly, then you pluck out the cellular bulb at the base of the hair, which has got rapidly dividing cells. We were looking at an aurora kinase B inhibitor, and we thought this was a way, by plucking 5 eyebrow hairs, by examining the expression and degree of inhibition of the enzyme in the hair bulbs, that would give us a measure of pharmacodynamics. Was the drug acting in the way that we hoped it would? But again, despite collecting the 5 eyebrow hairs per time point, the degree of variation was too large for us to be able to interpret. But an attractive concept of titrating drug dose to a biological endpoint. What about randomizing 2 different doses against each other? This could be done in a phase 2 trial, in which we look at some measure of efficacy and of course toxicity, because we would always want to give the least toxic dose, which worked, which was effective. A good example of that comes from our own experience using folinic acid. Folinic acid is a vitamin-derived cofactor that moves methyl groups around within the cell. And we showed many years ago, as did others, that folinic acid can synergize with 5-fluorouracil. Molecular basis of this is well known. Folinic acid binds to and stabilizes the complex of 5-FU and its metabolite F-dump with the target enzyme thymidylate synthase. It's a lovely piece of cellular pharmacology, very clear sense of the mechanism of action. What we did in our very large trial, 5,000 patients in the adjuvant setting, we compared high-dose folinic acid, conventional dose, 200 milligrams versus low dose, 20 milligrams, and we found no difference in efficacy whatsoever. So a tenfold reduction in cost, and yet still around the world, people conventionally give high-dose folinic acid, despite there being good randomized evidence, 5,000 patients, for goodness sake, showing that at a clinical level, at least in the adjuvant setting, colorectal cancer, it made no difference whatsoever. There are other good examples in phase 1 trials in which people like Mark Rattain, Ian Tannock have gone back and reanalyzed phase 1 trials and shown that we can detect efficacy, effectiveness at dose levels below the maximum tolerated dose. And yet, the groups involved in the trial continue to dose to the very highest level and select those as the drug treatment regimens that go forward, even though sometimes during the phase 1 trial, you can see that the drug is effective at earlier drug levels, at lower drug doses. But this is ignored somehow with that desire to treat almost to the point of maximum toxicity. This doesn't make sense, not in the modern world, I don't think. One other thing that we should consider is, how do we deliver the drugs? Do we really need to calculate the dose of our drugs with such precision that we use a multiplier of body surface area? The rationale for this is weak. As a clinical pharmacologist, I would want to be able to take account of our patients' hepatorenal function, the organs involved in metabolism or excretion. If we had measurements of those, then of course titrating drug dose, we do this with carboplatin, for example, but there are a few examples in which we titrate drug dose according to the function of those organs which are involved in metabolism and excretion. And therefore, I think we should be much more widely thinking about the concept of flat dosing, and to provide that dose within a single vial so that there's no wastage. Ian Tannock did some work looking at delivery of immune checkpoint inhibitors, and the waste, the amount of drug that's often left over in the vial when we try to calculate it according to weight or body surface area, one could save millions and millions of dollars just by flat dosing and using the entire contents of the carefully curated vials. The other area that I'm interested in is pharmacogenetics. We've done a lot of work using genome-wide association studies in which we look at germline genetic variants, often in the enzymes involved in metabolizing, deactivating the cancer drugs that we use in the clinic. And of course, if we could identify, using genetic techniques, those patients who were at high risk of developing serious, life-threatening side effects of chemotherapy, we could identify these patients before they're treated, and in so doing, we would be able to significantly reduce death rate, improve safety, reduce the number of patients admitted to hospital with serious side effects, and save the health economy money. It's very expensive for us as an oncology community to take care of cancer patients who are admitted with life-threatening toxicity. And again, we've shown this in our own work with 5-fluorouracil and genetic variants of an enzyme called dihydropyrimidine dehydrogenase, and a couple of other proteins that we've discovered in our genetic studies. So we have every patient in the United Kingdom has a genetic test before they receive treatment with 5-FU or capecitabine. And real-world evidence shows that we reduce the death rate from fluoropyrimidine toxicity, we reduce hospital admissions, and in our nationalised healthcare service, we significantly reduce costs. So these are some of the areas that we should be thinking about when we're trying to optimise dose selection for our patients. If I could ask you just to read this for a moment. This is a work of Mark Brattain that I mentioned a little earlier. He looked at a range of orally available cancer drugs, many of them tyrosine kinase inhibitors, the modern targeted oral drugs. And what he found was that by lowering the dose, a reduction in treatment frequency, the cost of more than half these drugs could be very significantly reduced, but without diminished efficacy. So exactly the point that I was making earlier on, that if we analyse the phase 1 trials very carefully and thoughtfully, then in a very significant number of cases, we could reduce the dose of the drug, maintain efficacy, and as Mark and his group worked out, potentially save remarkably billions of dollars. So for all those young oncologists, I know you get excited about molecular genetics. I know you get excited about immunology, but all of you, please remember the concept of dose. That's what we can control together. I'd like all of you to think a little about some good old-fashioned clinical pharmacology and some of the basics like flat dosing rather than the overcomplicated dosing regimes we use around body surface area. I do hope you've had a chance to listen to this brief lecture. I know that you're going to enjoy the webinar series, unquestionably, but I hope what this does is cause you to think a little about how we best control dose. Thank you very much indeed for listening.
Thank you very much, David. This is another pearl. And our role, all the audience today, and all who will receive these seminars and webinars and publication, everything, all of us should advocate for for this work and to be increased and to be adapted in any. The next speaker, I hope there will be still with us, Catherine Van Loon. Catherine Van Loon is a professor of clinical medicine in Florida, and she is director of a center in United States, and also she is the new director, chief editor of the very important journal, Global Oncology, GCO, which was founded. The founder editor was our friend David Kerr, who was in this board of editors. So her very promising role in GCO to give a big push to this movement of global oncology via science. Catherine, if you kindly present a few words about you, and then we'll have your video.
Hi, good morning, or I guess it's morning here in San Francisco. Very nice to have the pleasure to meet with you all. I can tell you a little bit about my path to global oncology. I was a master's in public health student in 2000, and had the opportunity during the course of my master's training and then at various time points during my medical training to spend time in South Africa. And my first visit and work in South Africa for my dissertation was at a time when antiretrovirals were not yet available, despite the fact that all of the clinical trial work around vertical transmission had been performed in South Africa. And so that's always been an inflection point in my career as we stared at a problem that at the time seemed really unsolvable. And the cover of Time magazine featured an image of a meteor descending on the continent. Over the course of the past 20 plus years, we've seen the HIV epidemic and landscape change dramatically through a series of pragmatic and stepwise changes, as well as with a foundation of international partnerships. I became an oncologist because I loved the patient care and chose a path that really was unprecedented. I was in the middle of my fellowship in 2011 when the WHO hosted its high-level meeting that declared the rising burden of non-communicable diseases as a new priority for the global health agenda. So perhaps a bit of serendipity, but I have had the opportunity since then to merge my passion for oncology with global health. And I'm thrilled to take on this new role as the editor-in-chief of JCO Global Oncology. And of course, I'm following in large footsteps behind David and Gilberto Lopez in this role. So I was asked to give a few words about how journals and publishing can impact value in global cancer. And so I'll let you play my video and then take some questions at the end.
Thank you very much, Catherine. And we count on you in spreading the message everywhere and leading this movement, co-leading this movement.
Hi, I'm Catherine Van Loon. I'm a professor of medicine and gastrointestinal oncologist at UCSF. I'm the new editor-in-chief of JCO Global Oncology. I'm going to speak with you today about the role of evidence in improving value in global cancer care. What is value? Treatment advances over time have resulted in very significant improvements in cancer outcomes in high-resource settings. Many patients, however, in resource-constrained settings do not have access to the same treatments due to access issues and high costs. Meanwhile, some treatments that we offer marginal benefits, which really need to be balanced against clinical toxicity, time toxicity, and financial toxicity. According to Common Sense Oncology, our goal as clinicians is to provide patients with treatments that provide meaningful improvements in the outcomes that matter the most. One of the areas that I'm interested in is strengthening resource-adapted guidelines for the future of cancer care in resource-constrained settings. Currently, most resource-adapted guidelines, including those put out by NCCN and ASCO, are derived from guidelines that are developed for use in high-resource settings. These incorporate very limited context-specific evidence in their development. Evidence that's generated in high-income settings may have limited generalizability to resource-constrained settings, and this lack of generalizability can serve as a major barrier to guideline uptake. Thus, there is a pressing need to strengthen context-specific evidence and evidence synthesis in generating guidelines for use in resource-constrained settings. These are a few examples of research that can improve value in global cancer care. For example, leapfrog technologies that offer timely access to diagnosis can be game changers. Point-of-care technologies have potential to replace slow, expensive immunohistochemistry studies. In precision oncology, we're evaluating the use of biomarkers to help us select which patients are most likely to benefit, thereby selecting patients who do derive value and improve outcome, and not exposing all patients who are unlikely to derive benefit. Multiple studies have evaluated de-escalation of cancer care, even in high-resource settings. For example, in HPV-associated oropharyngeal cancers, de-escalation of care has become the standard. In addition, in stage 2 and stage 3 colon cancer, 3 versus 6 months of adjuvant chemotherapy has been shown to be non-inferior and has been adopted as the new standard of care. In addition, evaluation of alternate dosing strategies like hypofractionation and lower dosing of immunotherapy have the opportunity to save substantial costs and time. As editor-in-chief of JCO Global Oncology, my vision is to enhance the literature with high-quality research from around the world that has potential to inform evidence-based cancer care in resource-constrained settings. To achieve this, I aim to increase visibility, views, and impact of global oncology research. I aim to promote equitable authorship in the global oncology research field that reflects the geographic distribution of where research data is generated. And I aim to expand dissemination of knowledge and reduce barriers for publication of high-quality research that originates from resource-constrained settings. Thank you for allowing me to be present with you today.
Thank you very much, Catherine. And we count on Professor Catherine Van Loon a lot in spreading and diffusing and leading the flag for the message of global oncology and how to increase affordability of better value care via science, via scientific studies, via evidence, and not by what happened along the last decades until now, slogan, meeting, speaking about disparities, about equality, and about all this. Our role is not speaking about disparities and to count, but our role, I think, if you agree with me, that what to do as scientists, what to do as oncologists, and to not to repeat what attributed to Stalin, that the death of one person is a tragedy, but the death of 1 million person is just statistics. So every cancer patient have a pulsating heart, have a family, have everything, and this is what is important, and it's not numbers and showing fungating breast or in Africa. This is the usual stereotype. This is signs of failure. If we present this is failure. And if we say that the gap will increase and will increase, this is a failure. So I think that the role of scientists and eminent persons like all of you to lead movement of scientific study focused on how to increase affordability. And I think the Global Oncology Journal of GCO have an important and leadership role in the near future, and we count on you and our colleagues. What do you think?
Yes, absolutely. And I think today's conversation has been really insightful, and the talks about how we make these next steps have been really very, very spot on in terms of where I think we need to go. And we spent the past decade, the first decade of the journal, I think, creating some noise, describing problems, understanding more about the global cancer landscape. But now is really the time when I think we need to shift into how we make forward progress in terms of care delivery with innovation and challenges to our traditional paradigms of how research is conducted.
We count on our continuous communication, and sorry for bothering you with many emails and many things, because there's a lot of work to do. And I'm just coordinator and just communicator with all of you, and we count on your leadership. We need a lot of leaders to lead this movement. Please allow me to give some outlines for the fourth and fifth webinars. It was supposed that we start with this, but I wanted to have the honor to listen from all of you.
I think also, Ahmed, while you're doing that, I think one of the important things I picked up from Catherine's talk in her editorship of GGO is looking at implementation science, something that Ian touched on, all of us really, the gap between the work that we publish, the lectures that we give, this webinar, and actually driving our ideas into real clinical practice and so on. And I think that the sort of leadership that Catherine will provide in that area and work that we can do in perhaps in low and middle income countries, I think could be really important. That gap's existed for as long as you and I have been oncologists, I think, Ian.
Yes, and I mean, obviously, we have a huge opposition. I mean, practice-changing trials, more than 90% are now sponsored and organized by pharmaceutical companies. And obviously, their main responsibility is to their shareholders. Sometimes there is cohesion between wanting to do the best for your patients and the shareholders, but that is often not the case. I think we have to start working with the people who can make a difference. I mean, the evidence is already out there, as we've heard from the talks today. We need to influence the people that can really push things. That's what we're trying to do in Common Sense Oncology. We need to influence the regulators. FDA is in a mess at the moment with the American situation, but influence EMA. We need to influence journal editors. In our Common Sense Oncology meeting in Montreal a month ago, we had Jonathan Friedberg, the editor of JCO, come. We had David Collingridge, the editor of Lancet Oncology. We had Nora Dices from JAMA Oncology online. So there is a movement to try to put stronger standards into pushing this. And the other place, of course, where we're trying to do things is through the World Health Organization. Of course, they've taken a big hit through loss of funding. The Essential Medicine List Committee is actually meeting as we speak this week. We had a cancer experts meeting in Geneva in January. And one of the things we're trying to get the EML people to do is to endorse lower dosing of particularly immune checkpoint inhibitors, less frequent treatment, for which, as we've seen, there's plenty of evidence. It's being taken up in low and middle income countries, but if the weight of the WHO comes behind it, it will make it easier and it will help people. A lot of people are frightened to give lower doses because, you know, not everybody responds, of course, no matter what you're doing. And there are a lot of people, particularly in the West, are frightened. Well, I didn't respond. It was because you didn't use the registered dose. So I think implementation science is absolutely key. We already have lots and lots of evidence. We should continue to get more of it. But even when we've got that evidence, as we've seen for the example of Septin, for example, I mean, it's amazing. Biosimilars have been accepted for treating adjuvant therapy of HER2-positive breast cancer very readily. The evidence to support biosimilars is actually considerably less than the evidence to support reduced dosing. It's amazing how people think quite differently. So we have a lot of work to do how to change those opinions. But ultimately, I mean, countries can't afford to keep paying more and more for these drugs. We want to have money to send our kids to school or mend the roads or things like that. So I think there will need to be a bit of a change that economic pressures will help.
I wonder, I mean, Catherine, just having Catherine here, another editor journal, is that something that GGO might get behind or consider attracting special articles or doing something around that whole interesting area? If WHO have assembled experts just now, I wonder if there's an opportunity for us, for GGO, to really think about how we could support that. I mean, obviously evidence-based and so on, but it could be timely.
Also, I convey the news also that with us, E-Cancer, with past editor-in-chief and patron, Eduardo Gazap, he is not with us today. He has the appointment with the Minister of Health of in Argentina, but he sent me a letter now. I'll read to you after my speech. And also we have Enric de Soto, the new editor-in-chief of e-cancer, and he will present his talk about the roles of e-cancer in diffusing this. We need a lot of that. We need a lot. And as you remember, David, when we have a publication and dissemination session during the Global Health Catalyst Summit and the win-win initiative for publication, David Collingridge was with us, and Ricardo Audecio and many other editor-in-chief was with us, and from Science and from many. And I send you the 1 hour record of this session, which is you can add to it your comments and your developments, really. So there is a lot of work. There is a lot of work, and there is a place for all initiatives. I see some revelries. No, we don't need revelries. It is a lot of work. The harvest is plenty, and the doers are fewer. So if you allow me to give this outline, and you notice that I speak quickly in this presentation because are a lot of examples, really, as Professor Antoniuk said, that there are a lot of things really proved and published, and as also Professor David Kerr said, and all of us, there are many things. And what is the really amazing, why it is not it is missed. If you see the upcoming conference of ASCO or ESMO or something, or what happening in low middle income countries also is not only in rich country, this part only 3-4% of the conference, if there is place, but all the other presentation.
Aman, can I show you one more slide?
Yes.
Are you here? Can I show you one more slide?
Yes, yes.
And I just want to take the conversation one step further in showing you this, because clearly you're all thinking about how to approach data generation. And I just want to show you this in terms of the pyramid of how clinical trial or how guidelines are generated. And we are using, unfortunately, the same pyramid that prioritizes randomized controlled trials over context-specific evidence. So just to push the conversation a little bit further for those of you who are involved with various panels for generating resource stratified guidelines, there is an opportunity here perhaps to shake this up and to move away from this very traditional pyramid where level 1 evidence is only randomized controlled trials. And in fact, we never see the context specific data and the data that's generated from diverse geographic settings show up as represented in generation of those guidelines. And I find that to be really problematic. This is a slide from one of my colleagues who's proposed a couple different ways to shake up the concepts of how we evaluate evidence in the global cancer space.
Wonderful. Wonderful, really. There's a lot of things to share and to and to do after together. Really, this is just a start, and we count on you.
Thank you.
Hello all. Today we'll start the ultimates of this series of ASCO GOIS International webinars. Today is the fourth webinar entitled "The Example of Scientific Exploration of Resource-Saving, Better Value Cancer Drug Systemic Therapy." As you know, this is part of BE certificate and of the program of BE Game Changers in Global Oncology. I am Ahmed Zawawi, Chair of Win-Win Initiative, also founders and president of ICDoc, and program of Global Oncology Implementing Science, is part of Global Oncology University, and Professor of Clinical Oncology, South University, Chairman of Al Suleiman Radiation and Clinical Oncology, Port Said, Egypt, past president of Aortic African Organization for Research and Training in Cancer, and co-president of SEM. This is based mainly on chapter 15. I am the author of the chapter, and the editor would be Will Negua, this book. Disclosure of conflicts, none. I completely follow scientific approaches, works of many scientists, and evidence. Moreover, we raise brainstorming, scientific stimulation, open for all what could lead to increased affordability of better value cancer care in the world, in the real world. I didn't invent anything here. I don't take fees for my lectures or financial support from any. I am aligned only to what could be beneficial to millions of patients with cancer, regardless of who they are or where they are. And I inspired this work for this initiative, and all what I'm presenting here is from the work of distinguished colleague, international organization scientists. So I invented nothing, and this initiative belongs to all. President of the whole series is Professor Robin Zone, President of ASCO 2024-2025, and here is a very nice introduction of her. The chair of the series, Professor David K., Professor Eduardo Gazab. You will listen today to their very precious lecture, really. Professor Antanok, very, very valuable lecture. Support your knowledge and our knowledge in such initiative. Professor Catherine Van Doon, Professor Hendrik Soto, planner and coordinator is me, distinguished border and panelist in alphabetical order, and here they are, and the porteur Jack and Abba. You'll find all details. Along the GOIS program, and it's this short lecture, we don't transfer some information in passive way, but we raise brainstorming points. So your feedback and positive interaction and following, and go, Sarah, the material here, and come back to us is very important. All your feedback is considered. The summary of some selective feedback will be appear in the next editions. We are partners. We are partners. As you know, despite of conference organization declaration of many efforts the world and hundreds of publications, health system would not cope with the skyrocketing rise of the total cost of cancer care, of that total cost of that do not commensurate with the increase of patient-centered outcome and affordability of better value cancer care. In the Win-Win Initiative, we advocate that the lowering total cost and increase better value cancer care could be improved with smart and scientific approach. All will win. No rivalries. We advocate to collaboration and close collaboration between all. The win-win is broader than just choose wisely based on views or surveys and not just following common sense. The Win-Win Initiative is a forum open for all and belongs to all. No rivalries. It is scientifically feasible to, this is an important question. It is scientifically feasible to lower the remarkably of the total cost of cancer care without compromising the outcome of patient, or is just talk? No, it rests on. Yes. It would be possible. True progress lies not in punishing the past and not in repeating the previous pitfalls or mistakes, but in fixing the future. This is what could be constructive game changers. And you, what do you think? A schedule of the fourth and fifth webinar with outlines and keynote speech you very valuable and precious. Listen to it, please, and come back with your feedback and discussion. Then will be open discussion, conclusion, and future direction. The record of webinars will be accessible to all. The PDF of these slides is available on demand. The next webinars, fifth webinar, will give more ample time for discussion and your feedback. So please go through this material till the next and the links that we will provide here. Material and links to the video of the lecture. Part 1, this is the link, and available in PDF. You will find here to click to find the details of this part 1. It's decreasing how to decrease the need of chemotherapy hospitalization. As outpatient, some known oral drugs, as you know, capecitabine, cyclophosphamide, vinorelbine, etoposide, and the oral hormonal therapy like tamoxifen, anastrozole, letrozole, exemestane, and progesterone. These all known. Example of new oral cancer drugs like oral taxanes. It is all details in the lecture. You will find it in the link here. Ongoing development of chemotherapy routes, subcutaneous, trastuzumab, pertuzumab. Another new exciting example of mode of administration is transdermal estradiol in prostate carcinoma. Transdermal. A new nebulizer device for treating lung cancer with the chemotherapy drug cisplatin. A pharmacokinetic study to changing regime of infusion. Starting this part 2, find the link here. The material here is a lecture that explains many things here. And here is the link. Starting with changing infusion regime, the phase 1 and 2 trial of infusion of gemcitabine are one example. Less frequent cycle of comparable efficacy, like zoledronic acid, Zometa, 4 milligram 12 every 12 weeks instead of 4 weeks in bone metastasis. Nivolumab, time is money, so 480 milligram every 4 weeks instead of 240 milligram every 2 weeks. This is according to Rattan and Goldstein. Atezolizumab, Goldstein and Rattan also searches for right dose and frequency by therapeutic drug monitoring. Other alternative is using standard 840 milligram dose, but with a dosing interval greater than the recommended interval of 2 weeks, every 3 or 4 weeks. Pembro, as you know, pembrolizumab, dosing every 8 to 12 weeks, gives similar and comparable results in non-small lung cancer. Shorter course, as you know, the famous example of trastuzumab and pertuzumab. Dose optimization of new drugs. A plea from Professor Marken Rattan and Alan Lichter to improve FDA, to empower, to support and to empower FDA for new actions for dosing, for adjustment, for revisiting the dose, and many indication and many things. Example of proposal of scientific studies that aim to lower the dosage while retaining comparable efficacy. Also, the FDA has done so for several drugs. The vast majority of oncology drugs over the last decades have been developed and approved at their maximally tolerated dose, which is not right, leading to off-target adverse events that may be fatal, as in the case of ibrutinib. Ibrutinib used in patients with chronic lymphocytic leukemia. Erlotinib, Tarziba, as you know, used in patients with non-small cell lung cancer, reducing the dose from 150 milligram to 100 or even 50 milligram daily. There are some studies outline. I present here titles, but there are details in the link here, and this it is the ultimate of our program and series. Presents example, and then you will wait from you after reading these materials. Dasatinib dispersal used in treat to certain type of chronic myeloid leukemia, low dose of 50 milligram daily, continue to demonstrate faster and deeper molecular response. Pembrolizumab in patient of non-small cell lung cancer, melanoma, bladder cancer, head and neck, and personalized dosing converting from flat 200 milligram to 2 milligram kilogram dose of pembrolizumab. Value-based prescribing strategies, this is very important, and that do not compromise outcome for new oral drugs alone. Alone, it could say, this alone could save up to 12 billions and more globally per year. This is better than searching for doing with what we have and looking for sense in reducing this. No, we can do many things in scientific way and not satisfying with this is what we have, then we give it is essential drugs and forget about the others. No, and it could save about $100,000 or maybe more per patient. After many publication, presentation, and plead and demand by many experts, including us, that we cited over years, you find the reference here in our books, we presented this. A great news, Project Optimus. It is by the FDA itself, but for its Project Optimus for reforming those optimization and those selection paradigm in oncology. It's very important. Reforming those optimization, all what is present in the guidelines in the last years and many things. Revisiting and reforming and optimization and those selection. However, according to Professor Ian Tannock, as you will listen today, unfortunately, the FDA will apply that correction for what will come and not for the previously approved. This, unfortunately. Do you agree with that? Professor David Kerr, optimizing dose selection for cancer medicine, a very important lecture. Professor Antanok, can anti-cancer drugs be administered in shorter course at lower dose to improve access to effective treatment? What evidence do we need to change the practice? Professor Eduardo Gazap, and also important relevant reference and material you'll find here. Part 4, you'll find the link here, starting with food effects then generics, then repurposing, part generics and part repurposing after. This you'll find all this in part 4. You'll find the lecture here. Example of those interaction using food effects and drug interaction. Lapitanib converting from 1,250 milligram with fasting to 500 with food. Abiraterone converting from 1,000 to 250 milligram with food or grape juice. Pazopanib converting from 800 milligram with fasting to 60 milligram with food. Certinib capsule converting from 750 with fasting to 450 milligram with food. Venclexta capsule converting from 400 milligram to 600 or 600 to 70 milligram daily only when taken with 300 milligram bazoquinazole. Generic equivalent and biosimilar of cancer chemotherapy. Links, there is the links. You'll find a very nice comprehensive approach here. Repurposing and repositioning. This is the first is the link to our first webinars. It's very interesting. A webinar of 2 hours is repurposing and repositioning drugs. This is one of the approaches, important approaches. Item of material of this webinar next time. But this material, please go through it and revise it. Starting with pharmacogenomic studies, pharmacogenomic study and value-based treatment. Evidence-based medicine could reduce cost and achieve equal or better outcome. More spending on cancer drug could have lower value. Reduction of the use of expensive supportive care drugs without strong evidence. Next, value-based reduction in overall therapy. Then, less toxic pathways. Part 6 is very important, starting with counterfeit drugs. This is the missed global campaign, the fight against counterfeit drug. You'll find this counterfeit medicine is worth 200 billion US$ and cause 1 million deaths annually in the world. Are there proposed solutions? Yes, yes, yes. And you will find some example of solutions in the text here. The full lecture and in our book. But you find here the link. Part 7, starting with control visualization of drug. This is win-win notion and initiative. The pharmaceutical response to interventional pharmacoeconomic study, the choosing wisely campaign and initiative, the other scientific initiative could contribute in reducing the cost of cancer drug treatment, previously taken in third outcome. But the win-win is broader and encompass and englobe all this. And based on scientific evidences. Repairing the compensation system used for oncologists in USA and in other countries, the rich and the less frequent, less affluent country. That we wanted to be this is if we want to be realistic in the real world. And there's final notes you'll find here in the lecture. Please note that I didn't invent anything here. It belongs to all. This initiative belongs to all. The scientific initiative will grow broadly and widely and globally and be globally effective only with your contribution. All of you. Advocacy. This is yours. And with the leadership of many scientists and oncologists and experts. Welcome all and thank you. Thank you all. We are waiting your feedback, and this initiative belongs to you and going forward in fruitful cooperation. Thank you all. Bye-bye.
Thank you, Dr. Zawawi, and thank you all for the excellent lectures.
I hear now Professor Sami Khatib. Professor Sami Khatib is very well known oncologist in the area and in the world, and he is guiding the AMAC. And you can give us 2 minutes about AMAC and then your opinion.
Thank you, Dr. Zawawi. Just as you said, I'm the president of Jordan Oncology Society, and I'm telling you hi from the area of the Dead Sea in Jordan. So about AMAC, it is the Arab Medical Association Against Cancer, which is the umbrella of 19 oncology societies in the Arab world. And we are doing a lot of activities yearly, and we are happy to be with Dr. El-Zawawi all the time. So if we can do any collaboration with any of our colleagues, this lectures is really, really, really excellent. And the reduction in the quantity of the money is, I'm hearing that is much more valuable that you have a country with petroleum. If we are talking about $14 and $16 billion reduction yearly in the medication only of chemotherapy. So this is a very, very, very important information for me. I heard about it, but I didn't know that it is so huge, the quantity of money saving that we can do. So we can let the rich countries to do whatever they want to do, but we as low and middle income countries, we should be careful and we should start applying these protocols and these trials.
Thank you, and please be the knight and the leader to diffuse this and to lead this among all the organizations connected to AMAC and you are really we follow all your work with great admiration, really. So from now you are the knight or the leader and raise the flag in with all this organization. Speak with its name, contact other with your title and with your name as a leader of this initiative in all the area. And with you, as you see, a big leaders, and we are honored that they are with us, as you see, Professor David Kerr, whom you know very well known, Professor Ian Tannock, these big brains that we count on him, and Catherine, and all. We have a very Eduardo Gazab and many leaders, Carol Ticora and Will Negua. We have many leaders who are all around you. Go forward, please, in the region and outside the region, and we are with you. Raise the flag. Go forward.
Thank you. Thank you. Thank you. Inshallah, we will do it. As you know, Arab Medical Association Against Cancer, AMAC, is covering all MENA area, not only the Middle East, also we cover all the North African countries. So we will do it.
I would like to Professor Eduardo Gazab. He have an appointment with the Minister of Health in Argentina, and he sent for us this short. He's, as you know, one of the co-founder of this initiative and co-founder of this program. He said that the National Agency for Health.
Yes, can you hear me?
Yes, yes.
Well, hello, Ahmed. It's to me an honor, and I am delighted to participate in this fantastic project, ASCO GOIS. So I would like to share with you only a few minutes to underline the relevance of public policy and regulations in order to improve cancer care and also to make treatments more affordable and more accessible for our populations. So I have 2 points to share with you. One is that, may I? So one is that Argentina is launching a national agency for health technology funding evaluation, a new autonomous body in Argentina that will assess the value and contribution of new health technologies, medications, devices, and including, of course, oncologic drugs. So this is an important governmental project that will, as main functions, will evaluate in a comprehensive and comparative evaluation of the current situation, including cost-benefit analysis and regulatory simplification. And an important point for our region is the reduction of judicialization of the medical treatments, mainly the high-cost medications, with the objectives of strengthening the economic sustainability of the healthcare system, promoting transparency and objectivity. We think that this initiative should be also part, as you know, the famous organization, agency in the UK, that is the NICE. This is something similar, and we think that without the governmental support, all the efforts to diminish the cost of medication and the access for treatment will be much more difficult to obtain. This is one example, I think, the actions that are taking place in our region to follow the principles that you are discussing today. And the second example, to finish, we are working with the National Comprehensive Cancer Network, you know, NCCN, with the guidelines, together with SLACOM. But the idea is not to adapt guidelines. The idea of this project that is going forward for around 2 years now is to make an evaluation of healthcare systems and how doctors in our country and region are following guidelines, and also how patients are following the guidelines according to the healthcare system's limitations and possibilities. So we think that at the end of this project, we will know better the obstacles and the favorable things that we have in order to make healthcare systems more efficient, more affordable, and more accessible for patients in our regions. That's it. I think that with these two examples, I can share with you and the audience some actions that we are taking together, medical societies and government, to improve cancer care in our region. Thank you very much for your invitation, and as you know, we will be in touch permanently to improve cancer care in our countries. Thank you, Ahmed and colleagues.
Thank you very much. And can I ask you something? Is as you are a patron of, he left. Okay. As you are patron of e-cancer, as we agreed together that e-cancer be a voice for this and spreading the message everywhere in the world.
I think that the idea is very possible. You know that today cancer is perhaps the only journal for cancer that accepts only papers from underdeveloped colleagues and regions. And I think that making, very well the current editor-in-chief, Enrique Soto, he is an advocate for all this, and I think that cancer will be very happy to take part in this movement.
So I hope that in the next 18 June session, it will be, it is the 5th webinar, this continuation of this, to have the talk that we have promised for it from e-cancer and how the promising role of e-cancer. We need all, in fact, we need Lancet Oncology, we need all. And our journal of Catherine, Journal of Global Oncology, GCO, surely. So I would like to hear from Jessica Williams, because she represents the ASCO committee here. Jessica, are you there?
I'm here. Can you hear me?
Yes, yes.
Wonderful. I just want to thank you for collaborating with us on this wonderful series, and we're looking forward to the future installments.
Yes, and it is an ASCO webinars. So we thank ASCO and thank all for this contribution, and this message will go forward only with the leadership of all, as contribution of all, and if everyone feels that it is his own. Thank you, Jessica, and thank you, ASCO, and all the staff of ASCO. Can I hear something from Dr. Samir? Are you there? Dr. Gianpaolo, and we thank you, Dr. Gianpaolo, really for diffusion of our message also. Dr. Gianpaolo. If any other comment, any of you would like to add or something, before I would like to also to give a final conclusion from you and from Professor David Kerr, from Ian Tannock, from yes, from Eduardo Gazab to everyone. Okay, you must leave, Eduardo. Okay, no problem. So, Catherine, you would like to add something?
No, thank you so much for the invitation to be here today. It's great to know that this group exists, and I look forward to additional partnerships with ASCO and JCO Global Oncology with this group. I'll definitely follow up about the idea of perhaps reaching out for a special article with updates to the essential medicines list. I think that's a great idea. So David, I'll send you an email to see if you have ideas about how to achieve that. And I'd say from the standpoint of JCO Global Oncology, we're always in need of reviewers. If you're not already a registered reviewer, send me an email. I'll put my email in the chat. And, I think my priorities at this point are really to publish things that are going
Malum.
to impact clinical practice. So we're really looking for rigorous research that has practice-changing implications. And if anybody has ideas for additional special articles or topics, I think some of the ideas that were thrown around today about rethinking clinical trial approaches are really particularly interesting. Increasing our focus on implementation science is of course a huge priority. And the other thing that I have been working on is also to improve ASCO's outreach to other regional and international global oncology groups and meetings. So I think we're unfortunately at a time where our international membership and attendance at ASCO is going to be threatened. Which means that the onus is really on me and on others to come to these global meetings and to regional meetings. And we would like to use JCO Global Oncology as a platform also for sharing conference proceedings. So if there is a regional meeting that's happening that you're aware of and would like to share content or keynote themes from that meeting, I'd be very interested in receiving that.
Excellent.
You said what I would like to ask you, really. So, Journal of Global Oncology, if we transfer it to first proposal that this fourth and fifth webinars, we have a rapporteur, Abba and Jack, and they write some draft. And if Professor David Kerr can lead this with you to appear in Journal of Global Oncology, JCO, that will be wonderful. And this is not just to inform people about this initiative, but to ask people to lead it and to be part of it, because it's progress only with that. Do you agree with that, David?
No, it's been a great afternoon, Ahmed. Thank you. And again, for bringing together such a fantastic cast of speakers. And I think, as Catherine has said, amplifying the sense that with the people who are listening just now, with the recording that we've made of this webinar, with the support of our friends and colleagues in ASCO, Jessica's here, we can amplify the work that we've done, we can amplify our voices, and to help to build the movement and capture elements of that which we've said, and see what we can do to continue to build in the momentum that you and others have created. It's been a really instructive, interesting afternoon. Lots of name checks for Ian Tannock, of course, for the work that he and Mark Brattain have been doing for the past couple of decades. But again, just to re-emphasize in a final point, that everything we do is based on science and evidence and understanding. And it's just the application of knowledge that makes a difference. And I think, as Catherine has said, it's not only the generation of knowledge, but it's implementation. And I think with the sort of group that we've gathered here, we'd hope to be able to drive change forward. So thanks again, Ahmed, for bringing us all together. Good afternoon, Ahmed.
Thank you all, and we need all your leadership, and we need all your pushing forward this movement. I think Abba we would have a last point to share. Abba.
Hi, Prof. Al-Zawawi and everyone. Thank you so much for giving the platform and bringing us together, especially for this very cardinal topic in terms of especially low middle income country. I think that's a very quite important topic. And above all, even before reaching to the clinical trial, I strongly believe that a clinician within the clinic, if we amplify this to different group on our day-to-day practice, we can be able to reduce a cost of chemotherapy or cost of systemic treatment in the clinic by applying the all method that are evidence already based, or either using a biosimilar instead of going for using a generic. And I believe that that will going to make a lot of cost saving or value for cost saving within the healthcare system and getting a effective and safety a better outcomes. And of course, as you mentioned, if opportunity were given on the platform of a global oncology e-cancers, I think this webinar series, we will be able to put it in form of black and white for a manuscript so that it will go beyond the boundary and beyond the border, and everyone will have access of that information. Thank you so much.
Thank you so much, Abba. You and Jack, we count on you and with me to write the first draft, and then all the distinguished contributors will add their point, and then will be raised to the final to David Keir and Catherine to propose and to make it in nice form in because we are not just chief editor, but you are leader of this movement. So you can add, you can modify, and for e-cancer for all journals. Thank you all. And if we continue speaking about what is this interesting point, we'll continue for hours. But and as we said always, this is not webinars for some tens of people or something and goodbye. But this is recorded and will be sent to hundreds of colleagues everywhere. And please encourage your colleagues everywhere and everywhere to apply for this certificate. It's not just for certificate, but to digest what is said and to add. And they are free to. We are not evaluation to say this is good or this is you are wonderful, but to say what they have of idea and even critics, even if it's constructive critics. Welcome all. And I'm really honored to be among you today. And thank you all. Thank you. And till our fifth webinars. And I am sorry to bother you with many communication, but because there was a lot of work. Thank you. And bye bye.
Thank you.
Bye
Thank
bye.
you.
Bye bye. Bye bye,
Bye.
Catherine.